So this is the new chapter and the very last chapter of the book, and it really talking about the very largest scales that we can think of ecology at, and thinking in terms of how do we scale up. One of the central questions being, okay, I go out to my stream or my lake and I measure this property. What does that mean for the whole watershed, right? What does that mean for all the watersheds in an area? And what we look for as scientists is the ability to predict what's going to happen in the natural environment as ecologists. And what we would like to be able to do is scale up our observations from the scale that we observe things at to the scales that matter. We've sort of touched on this in the other direction, and we talked about Reynolds numbers and Reynolds numbers in small scales, right? Where microbes are experiencing the environment at a very different way than we are. And so what is viscosity for a microbe relative to us? What is it? Higher, do they experience higher viscosity or lower viscosity? Higher viscosity, good, right? And they're dominated by molecular diffusion. Inertia is almost non-existent at microbial scales. And so that's the thing, that Reynolds number is actually a scaling factor that you can use to predict what the physical characteristics are depending upon spatial scales. This particular image is a mathematical representation of a stream, of a network, and they basically just used simple equations and some elevation, and they predicted what this stream would look like, how this would develop. And you can see it looks pretty much like you know, like any stream that you might see, except for it does look a little blockier, a little squarer, right? Because of the way they generated it. But you have first order streams and second order streams, and you have a lot of first order streams coming to the very biggest ones, right? And all those things based on fairly simple rules. And so this is part of the idea of these simple rules that we can use to predict stuff. I want to talk about hierarchies, refer back to some scaling diagrams that we had in earlier chapters and try to put these in context for you. There's a search for scale invariant properties, which I already alluded to, and the idea of emergent properties. So there's one of the things that one of my colleagues that when I started here at Kansas State University, he was a virologist, and he thought that the graduate students should just take biochemistry, and that they could figure everything else out from that, because everything was based on biochemistry. My response to him was, well, shouldn't they just take quantum mechanics, because everything in biochemistry is built on quantum mechanics? And the fact is that biochemistry is complex enough. It's much of it you cannot just predict straight up from the quantum nature of matter. Right. Maybe eventually we would be able to do that at some point. Similarly, you can't predict how an ecosystem is going to behave based on the behavior of a single organism or a few single organisms. And these are what emergent properties are, things that cannot be predicted that from smaller scales. The question is, how important are those? How are there things we just can't scale that we have to observe at the scale that they're occurring at? So this is an example of scaling. This goes back to the ideas that Luna Leopold, who's a hydrologist, had, and they're actually some of the ideas that led to the river continuum concept, that there are these properties of flowing water that lead to the shape of streams. And if you don't have these are scale invariant properties. And if you don't have a frame of reference, you can't really tell what scale you're at. And so the top image is a tributary of the Amazon River. It is a very, very large river. But if you look at the ratio of the curvature of these and how frequently the oxbows occur, it's proportional to the size of the channel, and that proportion doesn't change much. So here's a side channel coming into that, right? And the arrow now points to where we're at the next level, right? So we're just zooming in on that little bit right there. You can see those little blobs sticking out. That's that right there. And all these side channels and oxbows and things that are apparent here, you can't see them here because you're not looking at them. But when you get closer in this image, you can see there's an oxbow that's pinched off. So this will be a nice little lake and on the edge, right? And again, we've got this meandering pattern, right? And about sort of the same, you couldn't tell if you can't tell if that's 100 kilometers or 10 kilometers, right? Or 1 kilometer or 10 meters. And we can do this again. We can zoom in on this little part, this little nibble that's coming out here, right? And here we are. There it is, right? And we still have that same meandering pattern. And then we can see a lake, a small lake, right? That was maybe not so visible in the higher image. And out of that smaller lake, there's coming streams. And those streams are meandering at even a smaller scale. So this is the example of how scale can relate to hydrologic properties. And then you can imagine that those hydrologic properties are related to the biology of the system, right? This is what is creating habitat for large river fishes. This is what's creating habitat, such as we just saw in the last lecture on the Pantanal. And the fishing, the feeding the piranhas to the caimans was actually done on a boat ride that was on one of these little oxbow lakes. And that's actually the image behind me is one of those. So there have been several areas of ecology that have developed over the last number of decades. And one of those is called landscape ecology. And this is based on the study of spatial variation in landscapes at a variety of scales. And they include in the field the biophysical and the societal causes and consequences. It is interdisciplinary and is not necessarily scale dependent. So what they're interested in is how the spatial relationships on the environment influence the ecological properties. And one of the basic concepts of this is the idea of a metapopulation. The idea that if you think about populations of organisms, they can be spatially structured, right? That if you think about fish population in a watershed, there's areas where there's a lot of fishes, and there's areas where they're not, and there might be some habitat that's not suitable. So then you have to go a few kilometers away, and then you'll find another area, and those fishes interbreed there. The idea is that these form somewhat genetically isolated areas, and that the movement of organisms between those is really what drives what drives evolution in part, but also some of the characteristics of those populations. Well, we can think of this as source sink. Some populations are sources to other places if it's a really productive area, and other populations are sinks, whereas they can't reproduce at high enough rates, and so they basically are always receiving organisms from other environments, and then they disappear from there. This has been taken advantage of by some managers of aquatic systems. It's a really hot topic in marine biology, but also it's possible in freshwater biology. The idea, if you have a marine protected area where you have, say, a coral reef that you've protected, but don't allow any fishing on, then that is a very productive area, and then the fishes move out, and people can get the fish as they move out of the system. So it makes it for a sustainable system. The example I gave you of the Pantanal and Bonito, where they do not allow fishing for tourism, means that there's a lot of productivity of fishes, and then when you move out of that area to where people can fish legally, then those are basically source populations, and the ones that are being fished are the same populations. And we have many areas where we have regulations on fishes that you know, are just catch and release is allowed in an area, but if you move immediately out of that area, it's going to be producing fishes for that as well. This also leads to the concept of a minimal viable population, because it turns out that if you don't subsidize some populations, there's just not, it's not going to be able to keep itself going. So you can think of a population's distribution, and in the core of that distribution, there's lots of organisms, right? And there's a lot of genetic diversity. But if you get an isolated pocket out here, you can get inbreeding, you can not have enough gene flow coming into it, and it can sort of work its way to just oblivion, basically. So in conservation biology, you can't just get down to one reproductive pair of organisms, right? You have to have some minimum viable population. And these are based on metapopulation theory and understanding gene flow and how populations are being connected. This has practical application in conservation biology in freshwater. So for example, if you have, we talked about in western Kansas, where there's the stream network is drying up. And we'll talk about that in this lecture a little bit. And then you can take this idea to meta-communities. Are there actually groups of meta-communities that are sort of feeding each other? And it's a fuzzier concept, but you can imagine, kind of extrapolate that idea of metapopulation to meta-communities. So here's the example of the broadcast spawning fishes in the Great Plains and amphibian richness in prairie potholes. And the top one is work that Keith Giddo and Josh Perkin and his student Josh Perkin and some people have done. And the bottom one's one from the prairie pothole region in upper Midwestern United States. So in this case, broadcast spawning fishes are ones that put their eggs into the water, and then they have to have moving water, and the egg will work its way downstream and keep in the water column if there's enough turbulence. It just a little bit less than neutrally buoyant, and so it'd have to have active flow. If you run into a reservoir or in a dry spot, reservoir sinks, and they can't. The larvae won't be able to develop. They won't get enough oxygen. If you run into dry part, obviously, then it's just not going to work. And what that means is you have to have a network, essentially uninterrupted network, and you have to be connected. And so we have length of river fragment remaining in kilometers in the watersheds in the western part of Kansas and Oklahoma and Texas. And we see that if we can get up to about 300 kilometer fragments, we get about all of the species are hanging in there, right? They're all able to hang in there. But and then there's this threshold where if we get below about 200 kilometers remaining connected, we're not going to have any reproductive success. So there's this threshold. Similarly, we have the prairie pothole region in the upper Midwest. Anybody know what's threatening those potholes in the Dakotas, for example, in southern Canada? Yeah, farming, right? They drain the wetlands, they put fields on them. And I believe this one's from Nebraska, but yeah, I'm pretty sure that's right. In any case, so there's a regional species pool of amphibians, right? So if you have all the species that you can find in the whole region, right, in this wetland, then you're at 100%. So a few of them have all the species. You just don't find everything everywhere, right? So that makes sense. But if you have further and further away from a neighboring wetland, if you don't have this wetland complex where they're within 100 to 200 kilometers of each other, organisms are able to move from one to the other, and the source and the sink, right, source populations and sink populations, if something like this is just so far away that once the species go away, then none of the species are there, right? And if you're 500, 600 kilometers away from all the other wetlands, then you're not going to have very much richness, because once something goes extinct, it won't replenish itself. All right. The next idea we're going to talk about in large scale, and it's semi-related, is species area curves and the metabolic theory. Physical constraints of organism size, metabolic rate is controlled by temperatures, and predictions across broad taxonomic lines. So we've already talked about species area curves. Can anybody tell me what the relationship is between area and species diversity? Right. Good, Audra. More area, more diversity. More area, more diversity. Good, Morgan. Yeah. Yeah, so it started with island biogeography, but it could be lake size, it could be watershed size, in any of those things. What that means, though, is once you get a group of organisms, let's say you're interested in the number of fish species per lake, then if you get a whole assemblage of lakes of different sizes, you can predict how many species there should be in each of those lakes. This, again, comes back to the idea of that we talked about with the metapopulations as well. That if you put these two things together, if you need to have habitats, preserve habitats that are big enough to have a minimum viable population, and have them close enough to others that if they go down a little bit, then it can be replenished. This is part of the reason why we can't just preserve wetlands by saying, oh yeah, let's preserve one wetland every 500 square kilometers, right? Because when you do that, going back to here, right, we're going to end up with almost no of the regional species pool that existed there. And that's because smaller habitats support fewer species. And the examples we gave earlier in class had to do with the number of fishes in lakes, and it had also to do with the number of diatom species on slides, glass slides put in streams of different areas. And I believe also the number of species of invertebrates on rocks. And we did a similar laboratory on that as well. So you should be fairly familiar with that concept. Metabolic theory is another area of macroecology or macrobiology that has been developed by Jim Brown and his coworkers. And there's several things that are related to this. The physical constraints of organism size, metabolic rates is controlled by temperature, and predictions across broad taxonomic lines. We talked about, when we talked about molecular diffusion, and we talked about how organisms that are large are constrained by molecular diffusion, right? Because material movement in and out of cells is basically limited by molecular diffusion. How is it that large organisms get around that particular problem? Anybody remember? Like, how do we get around the fact that it would take days for oxygen to diffuse from outside of us into our center of our body? Where does a fish get, large trout has that same issue? What solution do they use? And sort of you get to the size of Daphne, and that's where the solution starts to kind of come up, where for organisms to deal with that. Take oxygen into the bloodstream, yeah, so how do they do it? What are they doing to get past molecular diffusion? What type of diffusion is faster than molecular diffusion? Nobody? Oh, the one that goes against energy flow? Yeah, you're getting there. So it's transport or eddy diffusion, remember, right? Flow. So what am I talking about when I'm talking about organisms? What kind of flow are we talking about in terms of getting oxygen to cells? Capillary, right? Circulatory systems. So what we're doing is we're using molecular diffusion to get oxygen, basically getting past molecular diffusion. We're getting red blood cells right up to next to the cells that need the oxygen with their hemoglobin, and then it exchanges, right? And so all the molecular diffusion has to go from the capillary into the cell that's right next to it. If it had to diffuse in from outside, we wouldn't be able to move at all. So that's a physical constraint of organism size. And organisms can solve those problems by having things like circulatory systems. And you can get to the extreme example of physical size being limited by the physics of being large. So the largest terrestrial organisms are not as big as the largest aquatic organisms. Whales are the biggest organisms, and that's because they don't have to physically support themselves, above land against gravity. So there's these are the types of physical constraints that we're talking about. That means is that you're going to spend more and more energy dealing with being big. If you're a big fish, right, and moving stuff through your body to get it to all the cells than if you're an amoeba that everything's diffusing in and out of. In addition, metabolic rates can be controlled by temperature. We talked about Arrhenius plots. We've talked about that several times, where you get this exponential increase in rates up to a point, and then when the proteins start denaturing, everything falls apart. So we have metabolism, and temperature is another thing that we have. So here's an example of the total metabolic output in watts on the x-axis, on the y-axis, and there's, what is that? Well, 14 orders of magnitude there versus the mass of organisms. So this just makes sense. You have a small motor, you can get only so much power out of it. You have a large motor, you can get more power out of it, right? We see that unicells down here are quite small, 10 to the minus 11th grams, and they can put out very few watts per organism, per individual cell. Large things like fishes up here and amphibians can be big, and they can put out a lot more power. However, it's falling off if you look at that. So I talked about the energetic constraints of being big. It takes more energy to be big and take care of that. So you can't have as much metabolic output per unit mass. How can we tell that? Well, look here, we're going from 10 to the minus 12 to 10 to the 6, right? So that's 18 orders of magnitude on the x-axis in mass between the smallest organisms that we've studied, the microbes, right, and the largest ones, a giant salamander or something like that, and big fishes. And then we go here, only 14, or well, maybe even less, 13 orders of magnitude, right? So we don't have as big of an increase in metabolic output as we do in mass. So that's part of the limitations of scaling to size to get at, what organisms do. If we think back to the ecosystems, right, we're thinking about how much power and energy it takes each level of ecosystem to function. And it turns out that unicells can be quite efficient, right, because they can put out more energy per mass if they have to, and they can use energy more efficiently than larger organisms can. So this limitation on the length of food webs can maybe be scaled back to the physical constraints on how organisms across a whole broad taxonomic range of types are able to deal with the environment.